So, okay. Well, welcome to everyone. I'm happy to introduce Martin that we have met last year in another talk here at UNAM, and now he asked me to give a talk here at the Institute. Apart from that, well, next week we are going to have a talk from Jimena. This is going to be more computational modeling, and next week is going to be more experimental. And well, Martin is going to talk about his PhD project, about how to model a single hippocampal nerve cell, and he works at the Interdisciplinary Center in Animal Research in Germany. So I think that is a different view from our research, and it's something that in Mexico is not so well developed. So it's nice to have this kind of talk, how to model cells and how to try to understand from a computational point of view how neurons fire and how the morphology and how everything connects. So well, thanks a lot, Martin, for being here, and we are happy to listen to you. And well, if you have any question, please ask me. Okay.
Nice introduction. Yeah, it was very kind of Roman to last year show us a bit his lab and the group, and this year I thought, yeah, why not have a talk and show a bit what our research is about. And I think it's two things. So first, this is kind of covering my PhD, so it will be distributed in 3 chapters. And what also Roman said already is basically computational. Most of the time, it looks like an electrophysiological experiment, but it is done in silico with a computational model, very detailed in the morphology and in the ion channel. So just to keep that in mind. Okay, so as a starting point, one can think what is a good model of a neuron. Quite often, right? You basically consider it as kind of a point entity that transfers input into some useful output. The neuron is the basic unit of communication and information transfer in the brain. So this is kind of a very typical and abstract model. And what this point neuron is doing in these type of models is to make a kind of summation, a weighted summation of the input. And then you have a function, I call it phi here, which transfers this into an output. It's often a nonlinear activation function. And you can have, of course, some learning algorithms that give you weights, how these inputs are summed, and that gives you parameters of this activation function. And then you can predict what the output should be. This is, however, when you think about the mechanisms that underlie the nerve cell, a bit of like treating the neuron as a black box. You don't actually know what's going on in this type of model. And the idea is in our work to make this box more transparent, to have an understanding of the intrinsic mechanisms such as ion channels and the morphology. Here's an example of a granule cell and how that affects the function. And in this case, for my talk, it's cells mostly from the hippocampus. Yeah. Okay, so as I already said, it will be kind of put into 3 parts, and that covers my PhD. And these 3 examples are examples of neural input-output function, and we will look at the underlying mechanisms that contribute to this function. First, I will have a look at what is called dendritic constancy. I will explain later in detail what it means. It is a principle that covers many cell types and sizes. The second topic is hyperexcitability in Alzheimer's disease. So hyperexcitability is one of the hallmarks of Alzheimer's cells, and we try to explain which mechanisms lead to the pathology. And the third project is pattern separation, and that is very important in the hippocampus, as the hippocampus is partially responsible for memory function and pattern separation, such as also pattern completion, important parts of that memory function of the book. Okay, these three parts, we start with the first. So the first one is dendritic constancy, which, as I mentioned, is an activity equalizing principle of the dendrites. And when you look at the kind of vast diversity among these cells, they have different topology, different shape. The architecture is different from the branching pattern. The size is different. You can have very small ones like the granule cells. You can have very big ones like these pyramidal cells here on the side. And it is quite intuitive to think, okay, yeah, the different shape transfers into different functions. And to a certain degree, that is correct. So here's an example. If you have a cell that has a very symmetric branching pattern, you stimulate it from the soma, and you get a certain firing pattern. The same cell, but with a very asymmetric architecture, the same size, same number of branching, but only to one side, and you get a different firing pattern, which resembles more like burst firing. And then in addition, we have many different computations that act on the dendrites and plasticity rules and so on. So of course, the dendrites matter, and the shape and the size of the cell matter. But in addition to that, you also have properties that kind of stay the same, no matter how you scale the cell or how you scale certain parameters. And dendritic constancy is one of these principles. So first, I want to give a little bit of a before coming to the results of these simulations, I want to give a bit of background, what type of modeling we do, and it's in general called multi-compartmental modeling. So you can see this granule cell here on the left. This is a reconstructed cell from mouse data. And then what the model does is to kind of make discrete compartments out of these dendrites in the form of cylinders. And you can have very small cylinder size, but you can also have a sort of coarse graining and very big cylinders. And in these cylinders, then certain physical electrodynamical laws apply. You can consider the membrane, the surface of the cylinder, which is a two-layer entity, as an electric circuit that contains of capacitance, and due to the ion influx and outflux of the cell, you have here these ohmic and ionic currents. This then sums up here to the capacitive currents. Here's one for potassium, for sodium, and the passive current for the leakage. G in this equation is the conductance, for the understanding. One example of this type of multi-compartment modeling is the model by Hodgkin-Huxley from the 40s and 50s regarding the giant squid axon. And the membrane or cable equation looks like this, and they could actually solve for these parameters of the conductances for sodium, for the leakage, and for the potassium by kind of doing a handwritten numerically analysis of the dynamics. They could solve this equation, and there is a lot in this equation. For example, this exponent 4 here resembles actually the underlying ion channel structure, because potassium in the giant squid axon has 4 subunits, and only when all of these subunits are open, then you have the influx of the ion or the outflux. Okay, and to apply, for example, this type of cable equation to this dendrite, you can then say, because charge is conserved and current is conserved, that the membrane current that comes in is then transferred to the next compartment, and you get this equation here on the bottom between these two compartments. In addition, we will always add, of course, synaptic stimulation, which is why there is also a term here for the synaptic. Okay. This is an example of a multi-compartment model. This is from Jarsig et al. 2005 for rat and mouse pyramidal cells. And you have here a couple of sodium channels and potassium channels here as well in the axon. And you can see that these channels are not always homogeneously distributed, but can have a length dependence. This is a very, very simple compartmental model. You can have more complicated ones taking in, of course, all the literature, all the experiments that exist, and then you have dozens of these type of channels, also calcium, of course, included. But this is a simple example of compartmental model that we put into a pyramid. Okay, this is the first in silico experiment. We have now these reconstructed cells. I think in total we have 100 hippocampal pyramidal cells. This is an example. And we stimulated synaptically. The synapses are homogeneously distributed. And then we see at the soma what the output at the soma is doing regarding firing rate. So we have several input strength of the synapses from 0.2 nanosiemens to 0.8 nanosiemens. Here you see the data cloud of 100 pyramidal cells. And the main message of this is that if you look at the scale of the size of the cells from small to big, that on average, the output firing rate is the same. And it doesn't matter how big the cell is, as long as the input density and also the diameters of these dendrites are normalized, you always get this average behavior, which is then what we call dendritic constant. Here on the right side, you see a stronger input of the input frequency of 2 Hertz. The input is always randomly distributed. I think it's a Poisson distribution to bombard these AMPA synapses with input. And yeah, and also in the stronger case, and here in the high and low conductance case, you see that the dendritic constancy principle that we state is holding. Then we were interested, okay, I mean, it's not always the case that the whole cell is bombarded with input. You have certain input pathways. And here we look at clustered activation, for example, apical dendrites versus the basal dendrites. Here in the middle, it's a subtree of a certain length, of a certain percentage of the whole cell. And here on the right, it's just, the color is a bit faint. It's just a cluster of synapses normalized to the size. And in all these cases, again, you can see for an input of 1 Hertz and synaptic strength of 0.8 nanosiemens here, 1 nanosiemens here in the middle. And here on the right, the input had to be quite strong with 50 Hertz because it was just a few synapses. In all those cases, the average behavior, the average firing rate is this. Next, when doing these experiments, we were interested in each step to add more non-linearity, to make the model more and more complicated. So we started with AMPA synapses. Here we add GABA and NMDA synapses. And in that case, when including inhibition, so here's compared black versus red, so only AMPA versus AMPA and inhibition, that of course the output firing rate is lower due to the inhibition, but the average behavior of the cells is constant and independent of the length. Below here, we add NMDA to the AMPA synapses for different strengths, for different input frequencies. And again, we can see compared to the only AMPA case, which are the black dots, the NMDA added in different strengths and frequencies and shifts the firing rate, but the average is constant for the whole data set of pyramidal cells. And finally, since this was now pyramidal cells in the hippocampus, but we were also interested if it transfers to other cells. So this is an example of cortical cells, cortical pyramidal cell from the layer 5. They are much bigger. And for that, we had to implement a different a different multi-compartmental model from Almog and Korngreen. The data set comprised of 20 cells, and in that case, again, with an input of 1 Hertz or 2 Hertz of background activation, you can see very nicely how the activity stays constant among different sizes of the morphology. Okay, that's for the first chapter. So basically, we revealed that there is a principle that underlies these dendrites, and it can kind of be considered as a backbone of equalizing cell activity, which we call dendritic control. We published this a couple of years ago in a paper, and the great idea is that this is a general principle, that it covers several cell types and orders of magnitude. And it kind of is active in the background, and on that principle, other dendritic computations and plasticity rules can then act on, which of course then have an influence on a very individual behavior of the cell. But as a background, this seems to be a principle for all the cells. Now, oh yes, I'm sorry.
The pyramidal cells, both in hippocampal and in the cortex, they have different distributions of channels in the dendrites, so they don't have a constant behavior.
You mean different individual cells have different sets of ion channels?
Not different. The same type of cell have distributed different channels along the dendrites. For example, they found out that principal dendrite has a lot of calcium channels, but secondary dendrites and tertiary dendrites, they don't.
Yeah.
And this behavior takes to the behavior of the cell, because the inputs in the tertiary one will be stopped by the calcium channels in the primary axis, while everything that goes into the primary axis goes directly to the soma. So this constancy that you are trying to show in the model doesn't check with reality. One is data, but it doesn't check with the McGee group.
It is simulation. So it is more, but I don't have it included in the main part. I can go to the end. There is we implemented a model from Poirazi. Let me just yeah.
They even show that if you put the inputs in different tertiary, in different dendrites,
I try
when they join together in a point, they can kind of step or go around the calcium channels that will block them.
Yes.
But if you only put one synaptic input in one of the dendrites, it will never go to the soma of the.
I try to get to what you just said. So in this more extended model, it's unfortunately a bit blurry. It's here in the bottom. It's a Poirazi model. It has calcium channels, T-type, L-type, R-type. They are differently distributed, as you say, if you take the main apical dendrite or the ones the more to the side. And when we investigated dendritic events, not only looking at the somatic firing, try to find here. This is a Poirazi model. The top is the somatic events, but here in the bottom is dendritic events. And that could be including calcium spikes. I'm not 100% sure, but it could be. Yeah. And there's a huge variation. Okay, but it seems to be on regarding if you want to make a correlation or something with the cell size, there is not. So you have it's true that these different inputs they interact with each other, definitely. But overall, the cell size, that's the statement, is not affecting it. You have exactly these events, okay, but the cell size doesn't really matter.
It depends on the timing.
That could also be because, so what we have here is Poisson input. It is
random.
But if you have a very specific input pattern, yes, I cannot. Yeah, that's
okay.
That is true.
I'm telling you about this McGee group.
Yeah. Mm-hmm.
Okay, and they are trying.
No, you're definitely right. So the statement is, maybe I have to bit carefully. So this dendritic constancy is under certain conditions, right? And it is like a background activation of this cell. But what you say is still then acting upon it,
right?
When you have specific inputs and so on. Yeah.
Well, they tried the input. They tried random and it didn't work. And tried synchronizing the inputs at different parts of the dendrite, and they found the calcium spikes. When the calcium spikes can cross to the apical dendrite, and when the calcium spikes cannot, will be passively submitted, blocked.
Okay. Yeah.
Okay. Thank you. Thanks.
So with this in mind, with this kind of general principle, we were asking ourselves if this could also be applied to pathological cells, where you have a change in the size of the cells to pathology. And this is now what we investigated in the second chapter. So the topic of the second chapter is the increased excitability that you can observe in Alzheimer's cells. So these are now pyramidal cells from the mouse. And the Alzheimer cells have this transgenic mutation of the APP-PS1, which kind of generates these amyloid beta protein plaques that are around the AD cells or the cells that are affected by it. And what studies have shown is that there is this kind of increase, not only in the firing, but also in the firing mode. So I show here like a table, it's a bit blurry again, but the increase is not in the single spikes. To compare wild type and Alzheimer's cells, the increase is in the bursts, which is doublets or triplets mainly. So the question is, what is the underlying mechanism for this behavior? The paper itself, where we also got the data from Zhishkova et al., made the connection that hyperexcitability is linked to dendritic degeneration, which is another hallmark of Alzheimer's disease, that the cells that got affected, they shrink. They show a retraction of the dendrites. And in this set of morphologies that we have, it's roughly 30 wild type and 30 with the AD mutation. You can see different parameters, the cell length, the number of branch points, and the surface area, that indeed the Alzheimer's cells are smaller than the wild type cells. However, our hypothesis first was that this is not the mechanism that leads to the increase in excitability. And the reasoning is that these Alzheimer's cells, during the progression of the disease, they also show synapse loss. And our idea was that the dendritic retraction to shrink the cells is a reaction to this synapse loss, to kind of get back to the same density of synaptic distribution. And then, in line with what we just discussed in the first chapter, with dendritic constancy, that would then bring up the activity again. So our idea is that dendritic retraction is actually a compensatory mechanism. Okay, and then we tested that. Again, we have AMPA distribution. We have wild type cell, the AD cell. And for several cases, this is, I think, the passive. Yeah, the passive case is here on top. The subthreshold active case with active ion channels, but below firing rate. And then here, for several input frequencies, we look at the average firing rate. And in all cases, the orange group, which is the Alzheimer group, is either equal to the wild type group or even below. So it's not showing an increase in excitability in these cases. And the main reason probably is that in the paper by Zizkova, that they stimulated somatically to test the hypothesis. And if you stimulate somatically, then the size of the cell, the smaller you have a cell, the higher is the input resistance. And with that, the voltage goes up. But that doesn't apply for synaptic stimulation, which is the more realistic stimulation that is going on in a circuit. And in that case, we don't see this kind of increase in excitability. And of course, the question is, what else is leading? What other mechanisms could be a cause for the increase in excitability? And we identified several scenarios. I will go into detail of each of it. And this kind of reveals that there is a multi-causal pattern mechanism, not just one. Okay, the way we did this is that at first, having the data from the Zizkova lab, that we defined an error function. Again, I'm sorry, it's a bit blurry. That compares the simulation results that we get with the data, especially with the bursts and with the triplets. And the closer we get, the smaller, of course, is this quadratic error here. And then we compare several configurations that are also known by literature that can occur during Alzheimer's. So what can happen is that excitation is increased, that you have a higher input frequency. It is known that the GABA synapses, the density is decreased in Alzheimer's cells. Then we have a combination of both, EI imbalance. We looked at the channels. So this looks quite easy here. It's just one line, but the investigation is quite tough because we have these dozens of channels and they interact with each other. And we try to do like a parameter search to see how they interact. And then, of course, also the channels interacting with the external mechanism. And lastly, we also check for burst input for a specific that the idea is that an Alzheimer's cell might show this burst behavior because the network is already showing burst behavior and it's just kind of infected by it. So we check for burst input also then in combination with other configurations. And in doing so, we could identify, so the low error is here in dark blue, 3 scenarios that we consider close enough to the data to say this is a plausible explanation for the hyperexcitability. And I will go through each of it. So in the first scenario, this is a combination of excitation increase and inhibition decrease. And the interesting thing is that on itself, excitation or inhibition changes did not lead to the hyperexcitability observed. It led to an increase in excitability, but not the burst excitability. Only in combination that the excitation is increased and inhibition is decreased, that we could see that the data gets close to, or the simulation results get close to the data. So it shows some sort of synergistic effect, the combination of the two effects. And if we then go through these panels, so here on the left is the general firing rate, and then we look at singlets, bursts, and triplets of these events. In gray, if you can see it, is what the data shows. So if you're close to that gray line, that is in accordance to the data. And then parameter 3, in this example, seems to be the closest match with the data, showing that from the wild type to the modified Alzheimer's cell, there is no increase in the single spikes, but there is a strong increase in the burst. So it's like nearly doubled. And you see also here a strong increase in the triplets. This is even nearly tripled. So this would be one explanation. Another explanation is the combination of this EI imbalance together with changes in the ion channel. Here on the right, there are 2 examples how we scanned these ion channels. So when we say we modify these ion channels, we mainly mean that the conductance of the ion channels is changed, because this is the parameter that stands for the distribution of the channels in the dendrite. And we can see here, for example, this is the sodium channel, that if you increase this, you get to a minimum. This is the after hyperpolarization channel. If you decrease this, now we look from the right to the left, you get to a minimum. And then we also check for combination of these channels. And on their own, only modifying the channels in the Alzheimer cells did not lead to the burst behavior. It also led to an increase in excitation. So we have an increase in these in the average firing rate, and we have an increase in the singlets, but not in the bursts. So only this combination of channels and EI imbalance then led also here again. This is parameter 3 with the best match. No, I think it's parameter 4 here. Yeah. With the data. Yes. And the third scenario that we identified is to look at the network. So you have input from the entorhinal cortex and from CA3 to CA1, where we are with these pyramidal cells. And if these are already infected with this bursty behavior of Alzheimer's cells, they can infect in CA1 area. So we started by basically having an input with majority of singlets and only one burst, which is a doublet here. And then we increased it step by step from parameter 1 to parameter 4, adding more and more bursts and less singlets. And in that case, parameter 3 then showed the best match with the data. So if you look at parameter 3, the wild type and Alzheimer's cells are very similar. If you look at here, that's a singlet. If you look at the burst, we reach a doubling of the burst, and for the triplets, again, a high increase with the modification to get to hyperexcitability of that. Okay. I already mentioned before that one of our hypotheses and confirmed was that the dendritic retraction, that the change in size of the cells, of the Alzheimer's cells, was a compensatory mechanism. And now we thought, okay, what if it didn't happen? What if we take the wild type morphologies that do not show this shrinkage and then apply these modifications that we investigated in these different configurations and see what happens then. And what you can see is, if you compare the green and the purple with the gray, that the increase of the firing rate, especially of the burst behavior, is even stronger. So one can argue that the shrinking of the cells, even though you see hyperexcitability in these conditions, it shows less than if the cell was still as big as in the wild type case. So this is an argument why the shrinkage might happen. Quite often it's considered a pathological feature, but we claim here that it's actually a feature of the system to maybe keep the homeostasis of the firing rate. Yes. And with that, I can already conclude the chapter 2. So we showed 3 scenarios of pathomechanisms that can, not one necessary, but none is necessary, but all are sufficient to lead to the increase in firing, especially in bursts in the Alzheimer's cell. And this is some sort of degeneracy. We always applied these modifications to the whole data set, but you can imagine that real cells, some cells rather show the scenario of increased burst, and other cells might rather show the changes in the ion channels. And still, these different cases then can lead to the same functional pathomechanism. That's why we call it degeneracy, which also makes it quite hard if you want to find like some sort of treatment for Alzheimer's cells, if we have so many mechanisms that could actually be the reason for it. So you have to identify what scenario and in what case fits, and then adapt your medication to it. Then again, the dendritic retraction that is observed at the same time doesn't seem to be pathological, rather helping the cell to kind of try to keep the firing rate as it normally would be. So a sort of homeostatic mechanism. Yes, we published this last year, and finally we get to the third chapter. So far we have looked at the firing of the cells. Now we also look at the temporal pattern of this firing, not just the firing rate. Yes.
When you check these cells, these shrink cells, does the input resistance
goes up,
get higher?
Yeah.
That will explain it, the perexability, just like that, because you have a higher, and so any input into the cell will be
that goes for somatic input. That is not for the synaptic.
Not for the synaptic input.
No.
So what you're measuring in the input resistance is only the input resistance of the soma, you say?
When we measure the input resistance, it's for the whole cell stimulating the soma.
Stimulating the soma.
Yeah. I mean, I don't have the data here, but it is increased.
It is increased. So it will be natural that anything that gets to the soma will fire.
I mean,
easily, will easily fire than that when the cell wasn't the normal case.
That was the argument from the Zhyskova group that also did some modeling, and that's where the data comes from. And they argued the same, that, oh, okay, but the cells get smaller. So yes, of course, they should fire more because of the input resistance.
But you say it's not sufficient.
It is under these conditions, yes, if you have somatic input. But the more realistic synaptic input that I'm
the synaptic
showing is, and the different pathways, also here from the entorhinal cortex and CA3, to the synapses, that's more realistic, and that does not show an increase in firing just because the cell gets smaller.
Okay.
Yeah. That's why we had to look for other. And that happens also, that all these conditions is not that I'm just, oh, let's change excitation. No, I mean, in literature, it shows that it happens during Alzheimer's, that you have a change in inhibition, excitation, and channel. So it's not out of the head. Oh, I don't know. This is the channel part is a prediction part. I can say from other experiments and so on that so these are two channels that I picked out of these four to show you here. I checked also, for example, for potassium channel. Potassium is also known if you change it. Actually, in the third chapter, I show adult-born granule cells, and these granule cells compared to the mature granule cells have a change in the potassium channel, the Keir channel, which increases excitability, plasticity. I could see that here, but keep in mind that we are looking here for an increase in excitability regarding bursts. The single spike firing, between the groups is nearly the same, is not significantly different. And with changing potassium channels and other channels, I could increase the excitability. Yes, I could increase the firing rate, but not the bursting. The bursting that you can see here is more than doubled, and the triplets even tripled. That was only for some channels that could lead to this input. Yeah. H channel also. I was so sure the H channel would do it, but no, not in this simulation. I don't know. I was reading. Yeah, of course, it's related to increase, but no. Yeah, this is the ones I could identify. Yeah.
Yeah. Oh, hello.
Hi.
Yeah, just in this slide.
So, which one? This one? No.
Yeah, this one.
Okay.
So you try many parameters, and just by the fact that the firing rate is not the same means that the error is not the same, and that means that you're going to have some error values that are lower than others. But that doesn't mean that the low values are good enough. So I was wondering, what was your criterion for saying, okay, this error value is low enough and the model is good?
Can you say again why a low error might not necessarily be?
Yeah, you're going to have high errors and low errors for different combination of parameters there. But how do you discriminate between like?
Yes. So
yeah,
this is a sum over different firing modes. So I look at the error of the singlets, of the doublets, of the triplets, and so on. And they are weighted because I know that the bursts and the triplets are more important. So there is a weight to these errors. And in addition to this error function, is not shown here, but it shows here, we look at those errors that definitely show a change in the mode of the firing. So you have the yellow shows that it's mainly single spikes, and the blue shows mainly bursts. And that in combination with the error, because you can sometimes find a lower error, but you still have more single spikes than bursts. So one has to, the error alone might not be enough. One has to have a look at it. But then again, if you look at the actual data of the firing rate, are you close enough to the data, which is this gray line? And yeah, in combination with this error function, one can conclude, okay, this is, I mean, what is good enough? It is in the overlap of the errors here, it is non-significant, the difference between the data and the simulation. Yeah. Oh yeah, that is absolutely true. Yeah, it's a local minimum. It could be, I mean, especially with the channels. I mean, I only have as much resources. I cannot change everything. I cannot look for a global minimum. I can say there are these tendencies. Yeah.
Martin, in the data, you can see a lot of variability.
Yeah.
Do you have an idea why this variability appear and how to model it? No. Okay.
No. I mean, this is very, when we go back to the dendritic constancy, here as well, you have a strong variability. But that's why we look at so many cells to make kind of specific conclusions. But why it is such a high variability? Partially, it's the diameter of the dendrites. So I try to use the realistic ones, but it is from the reconstructions of the cells is not always known, and that can shrink this variability. But otherwise, why also here with the burst behavior, why is such a high variability?
Change in time to an experiment. They get smaller.
And this is
they,
all.
They get,
I mean, this
they
is
put
all
spines. They get off spines. The dendrites are not static.
This is all simulation.
So you have a lot of problems here. Also
but this is not experiment. In that sense,
well, yeah,
I have my dendrite in the computer. It is fixed.
No, but the variance is just also due to the parameters you choose for the different channels.
Yes, I try to be to apply all these modifications like consistently to all the cells, and also with the ion channels. So it's the same set of ion channels and the same rules. There is variability regarding the distance-dependent channels, okay, but there is I don't know what variability you mean.
No, because
Yes, inside the compartment, it's not true. I mean, yeah, yeah. I can, yeah, I can here try to make these compartments smaller and smaller to get close to a continuous case, but obviously I cannot. It's not possible. Mhm. Yes. So I think I have a, yeah. I think I have a quite high resolution. I'm not sure how that explains the variability. Part of it for sure, but yeah. Mhm, mhm. Yeah, of course, it's an idealized shape here. So there is actually, you don't see it here, it's just a cylinder, but there's tapering. So I apply a tapering so that it gets smaller to the tip. There's tapering. There's a quadratic taper. Yeah. So in our group, they do, so I do mostly these multi-compartmental modeling. The other part of our group is trying to find general principles to simulate the growth of a cell. And in part is that they also look at these kind of how the diameters of the dendrites change. And there is some tapering and there is some scaling law involved and so on and so forth. So yeah, and that is in the model. So it is tapered.
Then you have source of
Martin, let's continue.
Yes.
No.
No, I mean, it's important. And yeah. I think we went to the third chapter. So this goes back to the initial slide that I showed you regarding input-output function. And now, not just looking at the firing rate, but to really compare the input-output regarding the pattern, looking at the pattern separation function of the granule cells. And we state that the granule cells are very efficient in doing the pattern separation. So first, a bit to the background. Degeneracy, I used the word already today. It basically means that you can have several sets of parameters that then still fulfill the same function. This goes also a bit to the ion channels that you talked about. So far, all the data sets, all the cells had always the same set of ion channels. That is not necessarily true. You can have a different expression of the channels regarding different cells. So there are very high individually differences. And that will be addressed here in this chapter as well. And then looking at the function of the granule cells, and the idea is that by evolution, this kind of parameter sets that you can find in the parameter space are reduced to the ones that are optimal for a certain function. And especially when looking at several functions at the same time, you can have an increase in the performance of one function, but it comes with a decrease in the other one. And in comparing these performances, we apply the perspective of Pareto optimality. That's how it's called. Okay, just to keep this in mind as a background. Now, what we have shown in a previous paper last year in our group is that the granule cell model, so this is here, reconstructed granule cell of the mouse, and the granule cell model is already a couple of years old, was also developed in our group, consisting of up to 15 channels. And if you have this variation in the expression of the channels here, you can still find different sets. So here are 4 different colors, 4 different sets that perform very, very similar. So when you look back in this picture, we have these different sets and the same kind of behavior. This was analyzed by our colleague regarding the somatic behavior. So we were looking at the action potentials at the interspike interval, at the amplitude, all these kind of electrophysiological parameters of the somatic action potential. And then looking at that, yes, you found one could find several parameter sets that perform very, very similarly. In fact, from 20,000 of these models, 6% were performing this way. The idea is that in nature, there again this kind of homeostatic principle. If you, for example, have due to some pathology that some of these channels might be harmed or something happened, that another channel could jump in and do the job to make sure that the function of the cell is still uphold. So that's kind of the idea, we think. And in addition, now for the specifically for the granule cell, we were looking, okay, maybe we could separate or constrain the population models that we have that all show the same behavior by looking at the economy and pattern separation performances. So what exactly that is, I will show in the next slide. But in doing so, we can get from one point in this function space to filling the whole space. And then by comparing each model with each other and only keeping the ones that cannot be outperformed in both of these kind of functions, we can construct this type of line here, which shows you the best models regarding these two functions. So I can give an example, right? You can find maybe the best model here on the bottom for the energy. It's very energy efficient. But as you can see, it is here in the upper left corner, but it is very bad in economy. It needs most of the energy. So in comparing these kind of functions, we can construct this Pareto line and say something about the performance of the granule cell regarding these two functions. Okay, this we can relatively fast. It's just to show you how we actually calculate energy. In this case, it's the ATP that is necessary for the sodium potassium pump to get the ions out of the system again. And we just do this integral over the currents and then have this relationship to the ATP. On the right side is a bit more complicated, and I will mainly tell you what the idea is. So in pattern separation, there are several measures possible how to say that a pattern is less similar than before. One measure is, for example, decorrelation. What very often is not considered in these measures is the content of information. So for example, if you compare input and output, if you compare input and output, if you had an output with no spikes, well, great. You have pattern separation is performed very highly because there's no pattern left. But it also means there's no information left. So it's not really a good measure. The decorrelation would be great and so on, but it's not really a good measure of functional pattern separation. So what we did was to include this information theoretical term, which is the mutual information, which basically says if you have the input, how much do you know about the output, or vice versa. It's a symmetric measure. And this is then multiplied with the so-called relative redundancy. This is the redundancy compared between input and output. And this looks inside the input pattern or the output pattern if there is high or low mutual information between parts of the pattern. Yeah, this is then our measure for pattern separation. Yes. Yes, okay. I edit it's here. I didn't edit here, but it's the same way. I consider the calcium currents, and it's so it's the contributions are coming from the calcium channels. I can directly. That's a great thing about the simulation. There is buffering, yes. Right, there is calcium buffers included. And I don't know how the buffers are included, but I mean, I include from all the channels. That's the advantage of doing simulation. I can look at the channels and I can see the calcium contribution. Also from the synapses, the calcium part that comes in, and so on and so forth. And I add up to get the charge. And then because of the ion pump, I think you need two. Yes, and that's why it's here. Yeah, so it's, yeah. I'm sorry, it's a bit blurry. Yeah, I didn't include it here because it's the same equations, but for calcium. So the contribution is here. Yeah. But yes, you're right. It plays a role. Yeah, so these are the two kind of tasks or functions of the cell to be economic, so use as little energy as possible, and to perform pattern separation as well as possible. And this is a simulation setup, right? We have a MATLAB toolbox developed to generate our input with a certain frequency and a certain phase and correlation. Then we have several trials that we feed into these yellow key synapses, these key inputs, and then we measure at the soma the firing pattern, compare both with the pattern separation that I just showed, and then get a number for the performance. And this is the result. So as I said before, we are considering here a whole population of models. So this is nearly 1,200 models that show the same somatic behavior. But as you can see, regarding the economy and pattern separation performance, you now get a data cloud. So they perform very differently. The triangle, that is the original model. That is the model validated by literature, published, and so on. And very astonishingly, it is on the Pareto front. So it is basically optimal for both of these functions of pattern separation and economy. It is nearly maximum regarding pattern separation and nearly maximum regarding economy. Not as quite, you can improve it, but then you get much worse in the other functions. So it's very astonishing that it is performing so well in both and sits here on these Pareto front. So the two colors are for two different inputs. One explanation in literature for high efficiency regarding performance is if the sodium potassium currents are separated in time. And intuitively, that makes sense because sodium is mainly needed for the generation of the spike and potassium then for the repolarization. So if you have an overlap of both, they kind of work against each other. So it is beneficial if these two currents are more separate. And indeed, what we could see is if you have a low overlap, you have high performance of economy and pattern separation. And this goes specifically, this correlates specifically with certain channels. It's a bit blurry again here. It's like KV, it's a potassium channel, one of the sodium channels. I think here is another sodium channel. So yeah, so there's this correlation between performance and the channels we need. Here we see it for the population models. On the bottom is the economy and pattern separation. And here the efficiency is putting pattern separation and economy into one variable by just dividing each other. And then we can see the original model here, these triangles, has the lowest overlap of these sodium potassium currents. Interestingly, you can see here these purple and pinkish data points. They were the models that also constitute the Pareto front. And interestingly, they cover kind of the whole cloud. So they could show low or high current overlap. But for the original model, that shows very low overlap, the lowest of them all, actually. Okay, and as a last topic, we were also interested in differences between the mature granule cells and adult-born granule cells, because in the dentate gyrus, neurogenesis is known as one of the rare areas in the brain where consistently during life, new granule cells can be generated, and they can also contribute to the pattern separation function. There are certain conditions different. We use the same morphology, but some of the channels are blocked, and that's what I mentioned before, some of the potassium channels, and the synaptic density. And then when we look at the performance, maybe we can concentrate here only on the left. There's another measure, but it's not so important. This is the pattern separation measure. The red color is the newly formed compared to the blue color, which are the mature ones. And you can see that they are kind of shifted to higher economy, a little bit less regarding the maximum of the pattern separation, but still they can perform the pattern separation. So the conclusion is that they can contribute to it with less energy even. Okay, we can skip maybe. Yeah, so to summarize this third chapter, we could show population of granule cell models that showed ion channel degeneracy. At the same time, this degeneracy could be separated or constrained when we look at energy efficiency and pattern separation function. And we revealed that the original model, compared to all the other models, is Pareto optimal for both of these functions. And in addition, when looking at the newly formed granule cells, they can contribute to this. A little bit as an outlook, what I'm basically have been working on the past weeks is to find out what the granule cell model is tuned for, what actual input. Because before I often look at this kind of distributed input, but of course they get a certain input pattern, as you also mentioned. And I look what frequency, what pattern they might be tuned for. Here there seems to be a difference between the adult born, which is this line, and the mature ones, this line, that there's a shift in the frequency tuning. And for the Kier channel, when you block it, there seems to be an optimal range for the adult born cells, but for the mature cells, it just goes, the performance just goes down. So this is like a little thing that I'm currently working on. And that's basically it from the talk. And yeah, I want to thank my supervisors, Peter Jelic, Bernd Hermann Kunz, from Frankfurt at Gießen. Some collaborations for the Alzheimer project with Laura Mediavilla and Stefan Remy. The current collaborations with Alexander Bird and the Della and Mongiat Lab. The Mongiat Lab in Argentina, we actually will fly there next month. Then a special thanks also to Jimena Salinas Hernandez. She will give a talk here next week. She's also important. If you're interested, yeah. The title is single cell model.
I know, but
no, but
you can network it, right?
I mean, I already in Alzheimer, I included network input, and one of the steps with the granule cells is also to have circuits of mature and adult born granule cells. So it is one of the next steps to have.
Okay.
Network.
So I don't know in person to Lucas, but well, he's at the same institute where I have been.
Yeah, I saw in Bariloche.
Yeah, but yeah, well, I left several years ago. So,
yes,
but
because he's working with adult born granule cells. I think actually now in fish.
Okay.
But before he also worked, we had data from mice from him.
Okay.
Some like 6 years ago or something.
Okay. Okay.
That's how it started.
So what are you going to work? Okay. So you are going.
I mean, I'm not 100% sure. The thing is that he has more insight into the features of the adult born granule cells to help us model.
Okay.
Yeah.
That's it.
Hi. Yeah, about last chapter, I think I kind of missed the original model that's optimal.
Yeah.
How did they obtain it again?
Right. So that's the fascinating part. No, it was a previous PhD student in our group, and it was a big literature research and many experiments to account for the behavior of the granule cells, what ion channels are included. It was not tuned for any of these functions here. That's why it's surprising. It was just, okay, what ion channels are there? What is the electrophysiological behavior? And then he built his, I can actually show you his model. I think. Yeah, here. This is a granule cell model. That's what he built. And he was just looking at the somatic behavior. And what we investigated then afterwards is for pattern separation and energy efficiency, it comes out of it naturally. It seems that nature is tuned to use little energy. Yeah.
So he just tried to replicate what he saw in, I don't know, in vitro or in biology, the best that he could, and then the best model arose. Okay. Yeah.
It's mainly somatic behavior. Yeah, that's true. Yeah, could be an upgrade. And in addition, I mean, I mentioned ion channel degeneracy. Again, this type of distribution can look very different in the next granule cell. We kind of apply it to all because, well, we don't know, but the whole idea of the population model was to kind of account for this degeneracy. So it's to then change this distribution and to scale it. So it was between 0 and 2, the scaling of the conductance.
Which program did you use for the compartmental
neuron?
Which one?
It's neuron.
Neuron.
Okay. And then we have an interface with MATLAB to mainly work on the morphology. It's called Trees Toolbox. It was trees, like a tree. Tree, which was developed in our group.
Any more questions? Okay. Well, thanks a lot, Martin. It was really nice. Yeah. Hopefully, this will open new collaboration, especially with Tonio. And but if any of you is interested in how to model neurons that you have at your lab, you can contact him. Yeah, it's really nice. Thanks a lot.
Thank you.